Okay. You go if I go live?
Yeah, man.
Hello, everybody. Welcome and good morning. We are out here. Here's Joe from Purple Martin Conservation Association.
Hey, chat.
You guys know him well. Also on the board for Alveus. We're out here in Austin. Joe, what are we doing?
What's up, chat? We're catching these guys. These are Purple Martins. And this is a female Purple Martin that is more than 2 days old. Or more than 2 years old. She's definitely more than 2 days old.
Nice.
And we are catching them and taking a lot of samples as part of our ongoing research here.
And you guys will see, this looks familiar to Alveus's setup.
Yeah.
But, oh, go ahead.
Yeah, and we were here last year on Maya's channel, if you'll remember. This is the exact spot where she hit me in the head with the paint pole.
I remember that.
Yeah.
That was wonderful.
Yeah, still suffering the effects of the concussion.
Oh, I'm sure.
But we're all good now.
But, yeah, Joe invited us out here.
Yes.
You know, not too far away from Alveus, get some experience with Purple Martins. And maybe next year when we get some Purple Martins at Alveus, you guys can come out and do some research.
We'll definitely come out, do some samples, maybe put some tracking tags on, who knows. Yeah.
Cool.
Sure, but, so it's in the bag, bird in the bird bag, and then I'm going to take it over here to where we are processing.
So what are we looking for today?
So we're doing several things with them. We're taking blood samples to look at their DNA to characterize a bunch of things, but migration timing is one of the things we're looking at with the genetic the genes that influence that. We are looking at parasites in the blood. We're looking at stress levels in the blood. We're looking at the overall health of the bird. We're taking feather samples to look at how much mercury they may be getting exposed to in the Amazon.
Dang, that's a lot.
A bunch of others taking just general measurements to just get the health of the bird itself. And a bunch of other stuff that I've probably forgotten.
That's so much.
Yeah.
Why put it in the bag, chat asks.
It keeps it calm. Calmer.
Do we have like a basket or something?
What's your tackle box? That was pretty good sticking
Sorry,
them in
yeah,
the lid of that.
yeah, if you could put that over there, that would be awesome.
Those at least keeping them from rolling away.
Wait, someone said wait till you see how they weigh the birds. That'd be cool.
Okay. I don't know.
They're interested.
She gets pooped on. This is Anna. She's from University of Central Florida.
Hello, I'm the poop master.
She's the poop master. I don't know if you want to be known as that online. Don't Google that, people.
Well, there's so much interesting stuff we can learn from the poop. I just collected it here,
Nice.
and so we're going to bring it back to UCF and extract DNA. And then we're going to sequence that DNA, and we'll figure out what insects
Oh, that's cool.
that birds have been eating. It is super cool.
That's tough to do, isn't it?
Well,
I mean, just trying to figure out all the pieces of DNA.
Yeah, of course, but there has been so much development in this technology recently, so we have the methods pretty well worked out.
Yeah.
Nice.
So we call it fecal forensics,
Okay, yeah.
figuring out what insects the birds are eating.
I used
So
to
this is
collect
pretty
raccoon poop
Cool.
for a researcher. Yeah.
awesome.
Yeah, so we used to have to pick through it and identify all the little pieces of insects because
Right.
they don't really digest the exoskeleton of the insect.
Sure.
But now we don't have to deal with the tediousness of that and the uncertainty. We can just plug it into the magic machines, and then it tells us what it's
crazy.
detecting in there.
I mean, when I took genetics, they were just really talking about doing that. They're trying to figure it out.
Yeah, it's DNA.
It's only been-
metabarcoding is what it's called.
It's been 6 years since they were talking about it, and they've already.
It's really
Done
come
that
on strong in the last couple of years.
Wow.
I'm gonna
That's
tell-
super cool.
I'm gonna tell the PMCA people that.
Oh, nice.
Very important to their well-being, to their health. So I've got this little swab in here. That one will be going back to UCF as well. I just took a swab of the cloaca.
Yep, okay.
So this we're also going to extract DNA from, but here we're not looking for insects. Here we're looking for microbial DNA to look at the
cool.
gut bacteria, which we know are super important for the health of the bird. So next thing we're going to do is flip her over. She's very cooperative.
Thanks, Sibby.
Going to take a blood sample.
Super duper egg. Subscribe. Thank you. Holiday memories back at uni. Okay. Nice, super duper. So, how much blood do you draw?
I actually take a very small amount. So these are glass capillary tube. It's just like a drinking straw. Same sort of action. So this will hold about 75 microliters, which is one big drop of blood.
That is so little bit of blood for such a huge amount of data. That's so cool.
Oh my gosh, it's totally amazing. And what's so neat about birds and reptiles is that the red blood cells have DNA in them. Human red blood cells, mammalian red blood cells do not.
I didn't know that.
So we can get a huge amount of DNA from just a very small amount of blood.
Why is that different? I'm just curious.
Why is it different?
Yeah, from humans to birds with them.
Over evolutionary time, that it's just been divergent lineages.
Okay.
And so dinosaurs likely didn't have DNA in their red blood cells either.
Interesting.
Because birds are dinosaurs.
Yeah,
They're
right.
living dinosaurs. So what I'm going to be doing is pulling the blood, putting it in some super special buffer that will preserve the DNA, so I can bring it back to UCF and extract DNA.
What's the buffer? Just curious.
It's a proprietary mixture that I buy from a company, and so what it does is basically preserve both the DNA and the RNA.
That's so cool. Okay.
So I'll do that. The other thing I'm going to do with the blood is make blood smears. And so I'll show you exactly how that happens, but they end up looking
Those are beautiful.
this. And so I'll bring these back to UCF, and I have a student that will stain them with 2 different colored stains.
Okay.
And we'll be able to see the cells much better. And what we're looking for there are the white blood cells instead that are part of the immune system. And it'll give us an idea of how healthy these birds are.
Okay.
So we make the blood smears.
Nice.
And then we're going to take a little bit of the blood and put this in our HemoQ instrument here.
Oh, that's so cool.
Going to tell us about the hemoglobin content.
Right.
The hemoglobin is what transports the oxygen around the body. So it'll give us an idea if they're anemic or not. So more health condition
It's
measures.
right there. That's so cool.
Yeah, and then a glucose meter.
Yeah,
So
okay.
I just bought this at the pharmacy.
Yep.
And we take their glucose measurements.
That's so
So
cool.
again, just to figure out the health of these guys.
You're doing like a full CBC chemistry profile.
It's a full workup of these birds,
Yeah.
exactly. And there's no copay for them either.
Yeah, that's
They
nice.
Yeah.
Yeah, so when you have a bird in hand, you want to make it count, because even though we're handling these birds as stress-free as we can, there's still some stress that goes along with being captured by a human and held, even though these guys are pretty used to people. We're trying to get as much science as we can to make it worth it for whatever stress those birds might be encountering.
No, please do. So I've got the wing outstretched here. I'm going to be collecting just a little bit of blood from the brachial vein that's right there.
So tiny.
So tiny. So I actually learned how to do this on house wrens, which are even tinier. So I'm just going to do a small little poke, and we get a little bit of blood.
That's easier than using a needle.
Yeah, capillary action. Pulls the blood up, get a little bit of a massage. There we go. And I don't even need a full tube. So as soon as I get that, I'm just going to put a little cotton swab and hang on to her for a little bit so that
That's so
clots
cool.
up. Isn't it? So I'm going to get this out of the way. So I'm going to make my blood slides. Actually, would you mind holding a bird? Sure. So we can't let her go yet. I just want to make sure that she's totally ready to go. So if you just hold one hand out like this, and then put the one on top, so it's just a sandwich. Okay? Perfect. So you just hang on to her for a couple minutes, and then I'll check her out before we let her go. So I'm just going to put a little drop of blood and make a blood smear.
Nice. I'm a vet, or a previous vet tech.
Oh, awesome! So
done a
you
lot
could
of
make
this.
these slides probably much better than I.
No, you're doing better than I've been out of practice for a long time.
Ah, just because I said that.
Yeah, sorry.
There we go. I think I may have.
Do you guys
saved
do
it.
manual CBCs with those?
Yes.
I'm so sorry.
I know. So I have a former undergrad student who was trained, and she's amazing at this. Her name is Isabel, and she's a full-time vet tech now, but she works for me one time a week where she comes in and reads the slides. But we're hoping to get a slide scanner next year.
I've seen those. They're
very
super cool.
expensive,
Yeah.
so I'm not sure if I can make it happen, but I'm hoping to be able to do so. And that will speed up the process a lot.
I've seen those, yeah, the machine learning ones.
Yes,
Yep.
there's one that can handle avian blood.
That's so cool.
Yeah. So I'm hoping that might happen. So this is a little cuvette. It already has reagents in it for measuring the hemoglobin. So I'm just going to pull some out. Again, it's all capillary action.
That is so cool.
And I'm going to open this guy up. So
Is this specifically meant for birds?
It is not. But a lot of folks do use it for birds. My brother happens to be an avian vet.
Oh, that's so cool.
He's in Vietnam. He was in Dubai before that, working on falcons.
Yeah, that's big money.
Yeah, so he was an avian vet there for a couple years, and now he's in Vietnam.
What's he doing there?
He's a vet there too. We're from Sweden originally, so
Okay.
they're probably going to move back to Sweden at some point. And so he decided to take a job there in Vietnam, a little bit of an adventure. And so he's working more on small animals now.
That's so cool.
Did you grow up in Sweden? Yes and no.
Thank you for the resub.
I was born there and then kind of moved back and forth.
Guys, if you're an avian vet out in Dubai, that is like the best place ever to do falconry.
Totally.
The amount of money that they pour into their birds and they treat them like royalty, it's amazing. It's a falconer's dream.
Oh, don't fly away yet. Yes, I had the opportunity to go out 3 times.
Oh.
It was so
You get to fly some of the birds?
I don't fly the birds. My brother flies the birds. Yeah.
That's cool.
So he actually learned that when he was a zookeeper at the National Zoo in Sweden.
Oh, cool. Yeah.
So he learned how to fly birds there.
Nice. We're flying a peregrine prairie falcon at Alveus, like a hybrid. He's missing an eye, so he's overcoming that. And then previously, I come from the American Eagle Foundation. We used to fly bald eagles and stuff like that. So
That's so cool. Yeah, I just work with the little ones.
I'm sorry, Joe's getting me into the little ones now too.
Yeah. Yeah, I started with the house wrens, moved on to tree swallows, and now I'm here with the purple martins, and I also work on Florida scrub jays.
Oh, nice.
Yeah. Those are a little bigger.
Yeah.
And bitier.
The
You want to let her go?
Oh, let's follow her chat.
Sure. All right. We have some picture action here.
Maybe a video.
Video. Oh yeah, slow-mo is the best. So make sure you're outside here so she doesn't get trapped up in the roof or anything.
Just take
so,
your
yeah,
top
go
hand
even-
off, and she'll
Just
jump
let her
right
go.
off.
There she goes. Thank you.
Join everybody else.
Oh, you got her mate right here.
Oh, really?
Oh, he's beautiful.
This is her. Yeah, let's go in the sun so they don't
Okay.
see the purple.
Set up at the very top.
Don't forget the bill. We have peregrines on the uni campus. Oh, that's so cool. Oh, he's feisty. Beautiful.
So they're definitely not purple. Hey, settle. He
Got
didn't
some spunk.
Yeah.
That's good though.
So you cover
Yeah.
them up, and they get calm. It's like
Little hood.
with people that put a hood on a falcon or throw a sheet over your bird cage.
Yeah.
If you got pet birds, same deal. They can't see anything. They don't know what they should be scared about.
When you go catch your next one, I'll follow you. I'm over here nerding out.
Yeah, no, absolutely. I mean, that's part of it. We missed out on some of the nerd stuff last time. So.
Did this couple have babies in their board? Or?
That's a great question. I don't know. They must, because to get them to go in while I'm there messing around, especially after I've caught a couple,
It would be unusual.
they would have to be motivated to re-enter the nest. And that's why this is the time of year where we do capturing, sampling, putting out radio tags, all that stuff, because if you try that before they have babies or after, they'll just sit there and wait you out. They don't have to go on the nest, but we know they have to. Like, they may hold back for a little while, and they're like, my babies need fed. So just instinctually, they will take a little more of a risk to go in there.
He just put a bird in the bag. They're hanging out here, keeps them calm in the bag. And once they're ready, they'll be taken over to the lab area here to get free health care. And then they'll be released. So for all the people that are just joining in, we're out here doing Purple Martin research with Joe from Purple Martin Conservation Association. All donations today go towards this kind of research and to this organization. And we have Purple Martin houses at Alveus, and hopefully next year, if we have Purple Martins take over, we'll do some research out there too.
Yeah, I'm feeling good about next year.
So she's grabbing blood, fecal, cloaca DNA,
feather sample.
Okay. Where do you grab the feather sample? That's cool.
So we grab R3. So if you're counting from the middle of the tail, I'll show you here in a second. It's the third one out, and if you're counting from the end, it's the fourth one in.
Okay. We've got to send off we have all the keratin and feather samples to send off to University of North Arizona.
Yeah, Arizona. We use the same guys as you do for sampling feathers. And tail feathers are made to come out easily, so it doesn't hurt the bird. They actually will pop right off, so it's like an escape tactic.
Oh, that's so cool. Yeah.
From predators, like when a lizard's tail comes off.
Yeah.
So if something were to come up and grab a martin by the tail.
Oh, that's so easy. Yeah.
Mm-hmm. I just brace it at the base.
Yeah.
So what I think is really cool too is you see these little like pin pricks there.
Mm-hmm.
That's feather louse damage.
Can we bring it closer to the camera by chance? Thank you.
So that's one of the things that my lab is really interested in, looking at variation and in the damage of the lice.
Right.
So.
Do you see stress bars on these guys sometimes?
Sometimes, yep.
Yeah, so and the reason we pull them instead of snipping them is that by pulling them, it stimulates a new one to grow. If you snip one, it'll just remain just the nub until they molt, which is grow a new set of feathers, and that happens with these all the way in Brazil. So these guys will have their tail feathers regrown or pretty close to it by the time they need to migrate. And
that's so cool.
It's good to have all your feathers when you're needing to fly.
Is that some poop?
Yeah, I'm gonna put that in a tube.
Nice.
Perfect. So, but now this fecal sample has touched a bunch of stuff, so I don't want to use it for microbial analyses because there's microbial DNA everywhere.
Thank you for the 20, guys.
Civi, thank you.
We will just use this for the insect DNA, and the cloacal swab will be the clean swab for microbial DNA.
45 grams. So you put backpacks on them. Are you just like tying string and doing like a grommet thing? Or
I'll show you on this guy.
Civi, you're awesome. Thank you. Trying to chat. We're working here. I'll show you guys what I've been working on separately from Alveus. This is an eagle tracker, 60 grams. It does everything that a $3,000 tracker does, but for $150. And we're going to see if we can take this and make it super, super small to 1 gram to fit onto these guys and more songbirds in general.
All right.
So how would we?
These guys get what's called leg loop backpack.
Yeah.
And it's like a backpack, except instead of over the arms, it goes through and under the legs.
Okay.
So each leg has a loop around it.
Yeah.
So up underneath the hip there.
Okay.
On both sides, and then it sits in between back here with the antenna facing backwards. So it sits low. And stays centered because when they're flapping, it keeps things pushed to the middle.
Thanks for the resub. Okay, so it's on the back towards the tail, and I don't think we'll need an antenna on ours.
Okay, yeah, no, I
Which is
saw
great.
that sample doesn't, and that's fantastic.
Yeah.
If you don't need an antenna, I didn't know how big it would need to be for.
I could be wrong. We might get in development. Might need a small one.
There,
but-
it is possible to have to put a tag up,
Okay.
an actual backpack over the
Right.
what are the arms of the bird, but it's
I like
less
that better
intrusive
down
and
there.
easier for them if it's down.
And I feel like that's more sun too.
Yes, if
down
it's
there.
solar, you're definitely in more sun.
Mick, thank you.
Joe inspired me to put up nests for the barn swallows on my run.
Nice.
That's so cool.
Barn swallows need
Now I
help.
have way less poop in my barn and horse stall.
Way less poop in his barn and horse stalls now.
Perfect.
That's so cool.
That's great. Yeah, barn swallows are just as important. They're undergoing the same population declines that purple martins are. They're great at eating insects. And the reason that we study purple martins and not barn swallows is, due to a couple of reasons, because people have such a close relationship with purple martins and putting the houses up. But also, they're really easy to do research on. We get a bunch of nests all in the same place,
Oh yeah, true.
and they don't mind us interacting with them. They're used to it, and we know that they're going to come back to the same place year after year. So if we put a tag on them, we know where to go get that tag the following year.
That's so cool.
How many did you band here last year?
It was a lot.
Don't
It was at
recall.
Least 10, I think.
Yeah, it was creeping on 20, I think.
Yeah.
And while that's a drop in the bucket to the number of birds that are here, it's for the amount of time it takes to process each one. We can only do a certain number in a day, and then we've got all these sites to hit all across Texas. Because where we're sampling is the same places last year that were affected by the big freeze in Texas that killed all the purple martins at that point, and we asked people to keep those birds and preserve them, and then we went through and collected them. So those birds are a genetic sample from migration up to the point where that freeze happened last year. And every single purple martin that was here at that point died because it's so cold for so long.
Jeez.
And then any birds that are alive now are the descendants of birds that came after that freeze. So we've got a different genetic pool from that. So we sampled the dead ones, we sampled the live ones, and we can look at the genetic difference between those two populations.
And it's a very unique
Yeah,
sample. This
that sort of thing doesn't happen. You can't make that sort of sampling happen on a wide scale like this. So if we were
Is that a bottleneck? Is that what it's technically called?
potentially,
Okay.
yeah. I don't know the specifics on what exactly needs to happen for that, but yeah, it's absolutely a trimming of the genetic diversity, at least when it comes to migration.
$4.20. Let's go UCF.
420, yeah.
Thank you for the 420.
But
I'll follow you, Joe.
nights, right?
Yeah. Although I feel like we should change it to the purple martin.
Well, Anna has installed purple martin colonies all across UCF campus. So I don't
Nice.
know if anybody is watching that's a UCF student, but
I've seen Disney's doing a lot.
Disney has so many. They're a wonderful partner to us. They've helped fund research. We work with them on so many projects behind the scenes.
Yeah, guys, Disney is big into conservation. How cool is that?
They're great. They're so great.
All right, Joe, walk me through what you're going to do.
All right. This is what you're going to do, actually.
Well, I'll watch. Well,
Okay, you want to watch one,
Yeah.
and then... All right. So, we've got a official purple martin. Did one just go into that?
I don't know.
Chat? Did one go into there? I don't know.
Oh, you got a
I
dragonfly.
think one
That's
did.
so cool.
Yeah, so they're feeding babies.
That's huge.
But the official purple martin catching apparatus, which is a extendable paint pole, we just sit here, let them get used to us, get their trust a little bit, and then betray their trust immediately by quickly putting this in front of the entrance when they go in, and then we lower things down because purple martin colonies are ideally on something that you can raise and lower to keep the sparrows out, and that, you can lower down and check on how nesting's going. You can remove the invasive species nests that are trying to take over. So we keep it blocked, we drop it down, reach in, grab the bird, put it in the bag, boom, process it, and then on to the next one. And yeah, it's really high tech. Sometimes the easiest solution is the best one.
Nice.
You know, we could set up remote triggered traps.
Oh,
and
yeah.
things on strings and this and that. But
Maybe we got something going in 19, maybe?
yeah. So that looks like a subadult male. So that's a baby, a male that was born last year. So you can look at them as kind of like inexperienced dads,
Okay, yeah.
but they don't. You see, it's kind of causing trouble up there. They're kind of bullies. They'll cause trouble at the colony when they show up. Break up happy homes.
Jeez.
They're, you know, the younger, inexperienced parents aren't that great at feeding their young. They're not that great at learning how to build a nest. So it kind of takes a year for them to get up to speed.
Right.
And it takes an extra year for the males to end up in that full purple color that last one.
So 2 years total to get that purple color, right?
Yep.
Nice.
So these are called subadults, which, you know, they're adult enough to breed,
Right.
but they're inexperienced. And the reason they don't call them 1-year-old birds is because when they arrive back, they're actually not quite one yet, right? So they arrive before their birthday. So you don't just refer to
Right.
their age, you refer to
That's
how
easier.
many times they've been back
Right.
from their migration. So, subadult, or SY, means second year, because it's their second year on the breeding.
Go in 22. Do it.
Do it. And then after that, they're ASY after second year.
Okay.
And we don't have any other names for them because they always look the same after that, right? It could be the third year, fourth year, fifth year, whatever.
Subadults, just like a streamer. Yep.
Exactly.
100%.
Exactly. And probably as good with the females as a streamer.
Yeah.
You know?
100%.
Roasted. Get roasted.
All right, 17 or maybe 17's in.
So usually just...
Need me to lower it down?
Yeah, go ahead and crank it down. So, you know, once they see something at the entrance, they'll just kind of get back to the back of the compartment.
Lowering it down, chat. How fast can I go?
You can go faster.
Okay.
Yeah, just watch your head as it gets down there.
But you have to get revenge on Maya through me.
There's plenty of time. All right, that's good. You want to come and grab one of these bags?
Bag. Okay.
And then go over and slide it into the entrance. Just kind of stuff it in the
Oh,
entrance
yep, I saw
in
the-
front of the paint roller.
I saw you do that.
Dad, did you see that crow just buzz right through there? It almost hit me in the head.
Nope. Oh.
I know, it's a blackbird. It's coming in hot.
Stuffed. That was a rap reference, wasn't it?
Yeah.
That
It was.
was nice. I like
Thanks.
that.
If you guys will remember, rap is my thing.
I didn't know that. What?
Oh my God.
Me too.
I love rap so much.
Joe, you're so hip.
Look, still got food in his mouth.
That's funny. Nice.
And you can,
Is there babies?
yeah, little bitty babies. Let me show you.
I can. Oh. Oh my God.
These are really young.
Oh my God.
They probably hatched yesterday.
Crazy. Oh, can I take a picture?
Yeah. And look down at their belly. See how their belly's so bulbous? It's still got the yolk from the egg in it.
That is so cute.
So small.
Very.
Eyes are closed for quite a while. Yeah, they just kind of...
How many do they normally have in here?
Take a look. I mean,
Oh, okay.
typical is, I don't know, 4 or 5. But they can have up to 7.
Oh, there's a bunch of them.
Yeah, it felt like a good pile in there.
Look at that, chat.
You know, and then down to obviously as many as low as 1.
Thanks for the raid. Hope you had a good stream.
Nice.
I'm hoping I didn't disconnect the camera. Nope, I did not. Okay. So cute. Okay.
All right.
That's awesome.
Yeah, so those are little bitty babies. That's why he had very small food in his mouth. They adjust the size of the food to
Right.
how old the babies are. And it's actually not very long until they eat entire dragonflies and giant beetles and horseflies. And
That's so cool.
purple martins eat so many insects every year. The species as a whole eats 250 billion insects every year.
Holy hell.
Yeah, and that's, we've lost a third of all purple martins in the last 50 years, so, you know, a third more than that,
Right.
whatever the math is, 50 years ago.
Jeez. So insects on the rise, unless something else is taking over.
Unfortunately, insects are also on the decline, which has conservation implications for purple martins and being able to feed their young, in part to habitat modifications by humans,
Right.
climate change, pesticide use. Pesticide use, we think, is probably a major factor in the insect decline, as well as climate change, changing the rainfall and the temperature and everything. All right, I'm going to give this to you. I'm going to crank that back up. You go deliver this guy.
And what they asked, what do you do with the bags after? You
We wash them.
Okay.
We wash them. We try and watch yourself. Yeah, we give them a wash every night, bleach them.
Nice. All right, chat. We are to deliver this beautiful male to his free healthcare checkup.
Underscore Peppermint just
I
subscribe.
did a volunteer position. That's how I learned how to do mist netting. Thank you so much.
Thank you. Is there still one in here too?
There is still one in
Okay.
there. That one's going to be next.
Okay.
So I'll
Oh,
put
cool.
this guy
I'll put that.
here so I
Awesome.
know that the one that's been held the longest will be.
They're now getting. We're referring to you as doctor for free healthcare.
Okay. Free healthcare. Excellent. Well, I only work on birds, so that's the only advice I can give.
This is very cool. Thank you. I watched the stream last year,
Yeah?
so it's nice to be out here.
Awesome. I'm so glad that you're here. Yeah, this is a lot of fun for sure. And this is such a beautiful place.
It is.
To be
It's nice.
sampling birds. And
Yeah.
an opportunity to meet lots of people that are interested in birds and the purple martins specifically.
Do you just fly up here for a few days and then fly back?
Yeah, I flew into Dallas
Oh, okay.
on Saturday and flying out from Houston on Saturday.
Oh, okay. You guys are making the rounds.
Yes. Absolutely.
All right, we're going to find Joe.
Back in, so I know where to reach.
Right.
And then
You
they
don't know
definitely
which
feel
head-
different.
Which way their head is.
Yeah, sometimes they come out upside down, but I can adjust it pretty quick.
Cool. They like your headband, by the way.
Thanks, chat.
You're stylish.
Fashion is important to me.
Yeah.
You know, you got to look good to feel good.
No, not true.
I got to get some Alveus merch though.
Oh, I've got to send you the. I'm sending you the posters. Yes.
Dude, I'm so psyched. Those posters.
They're
I don't
cool.
know if anybody's watching that made those posters, but they're incredible. I'm so jealous of anybody with artistic talent.
The Kayla, I didn't think she would like the. I think it was called Scum Art. That
Yeah.
one. Yeah.
Yeah, I mean, I love
She
it.
loves it. She's like, we're putting it up all over our house.
It's so cool. Like, I love, you know, on Twitter, the stuff that he posts.
Yeah.
Just the kind of warped angle on it.
Yeah.
Artists are so cool. All right, Connor. You ready? You've been trained.
Why are we outside of Alveus? Because we're doing research with Joe, and this kind of ties to Alveus because Alveus has. We're gonna do this next year, hopefully. Joe's also on the board for Alveus.
Yep.
And yeah, all right. So we're paying attention.
Yeah.
See which one is gonna go in a gourd. Is that the technical term?
These are artificial gourds. They are crazy looking because of features people have added to make them more predator resistant and all this and that. But really the whole gourd idea is to emulate what Native Americans would have used to attract purple martins back prior to European settlement.
That's so cool. And they wanted to attract them to help keep down pests and stuff like that.
Honestly, we don't know. It was a tradition that was established in a lot of Native American communities prior to Europeans coming over and writing about it. They just kind of showed up and we saw what was going on. Certainly insect control could have been a reason.
Right.
They can be kind of scarecrows.
Oh, cool.
Scare off, the crows or hawks, that sort of thing. They'll kind of go run them off.
Right.
They could have been like watchdogs. This sound you hear,
yeah. If something comes in,
that's like a warning.
Right.
A warning call.
7, maybe 7. Look, you got food. We going, Chad? We doing it? Come on.
7 or 10.
7 or 10.
Or 19. If you're good, you can
usher them in.
You can ones that you can go up
on
and around
the
back.
Feel like 7's showing like he wants to do it, or she. Is that he or she? I can't tell.
It's a he.
He. Okay. There's lots of purple. Well, bluish purple.
So the feathers aren't actually purple. It's what's called a structural coloration. So the feathers are really black. There we go. 7.
Yeah. Where's the hole?
All right. I'm gonna grab the bag and lower it down. Nice work with the quickness. All right.
Hopefully.
So as we lower it down, you have to kind of adjust.
And that's where Maya messed up.
Exactly.
I mean, I guess there's worse things to be hit in the head with, but
Yeah, no, it was all right. I've got a hard head. No worries.
all right. Let's see if I successfully got this guy.
You want to reach in and get it too?
Sure.
All right. Technique. Unscrew it and then slide your hand in. Just go as wide a hand you can go. And then the other hand, just go through the gap there. And it'll probably be towards the back.
Oh, chat phone. Chat, you're in the way. Let's trade hands here. I don't know what I'm feeling.
If it feels like a bird,
Yep.
you're holding onto the right thing.
You were very... Oh, there's another one. Is he dead? Or protecting? What? What is going on? Oh, these are
the babies.
These are babies.
Older babies. That's why it was hard to grab.
That was... I felt so many things.
Look
and
at
I was
these
so
guys.
confused.
Oh, they're big.
Yeah, these are good size ones. They still probably have
That's so cool.
10 days to grow.
Look at you. Say hi to chat. You're so pretty. Okay.
All right.
You got him?
Grab her.
Her. Okay.
Her. Okay, we didn't catch her yet. That's good. She's going in the bag.
So they do stress out a little bit with this, but they're pretty quickly released. And
Yep, we go as quick as we can. We try and keep them as calm as we can with the bags, and just if you hold them too loosely, it's actually more stressful for them.
they're expending energy that they don't need to.
Yeah. If you hold them securely, but not obviously too tight or anything like
Right.
that, you're actually minimizing the stress. And it's something that just comes with practice and knowing how to handle wild animals.
We're in North Austin. We're probably 20, 40 minutes away from Alveus. Thank you.
Right there.
And we're learning how to do this so we can do this at Alveus next year. Joe will come, of course. We'll be hopefully a part of this research trip next year. I just want to show you guys what's going on. All right, I will move.
Oh, perfect. To
This
the slot here.
Yep.
Thank you.
Good system. Good system.
Got a beautiful ASY male here, an older male.
Nice.
Undergoing his.
Chat, did you know a cloaca is where everything enters and exits? It's one hole for everything. So
And there's lots of microbes.
Urine,
lots of bacteria.
Poop. Breeding.
Eggs.
It's so cool. Eggs, yeah. Will they be for food? Nope. We are researching. Just researching. They're not Orion food.
How's the supply going?
We're going. We need a little bit of a break.
Okay, perfect.
I've got this one, then I've got 2 ones in queue. So.
Perfect. That's what I was thinking.
That's cool.
Yeah, this is a super. I love like public garden spaces and urban areas for people who don't have backyards or whatever. And this one's associated with the School for the Blind, right?
Mm-hmm. We're on their property. This is their land.
Got it.
So we're at the back of it.
Yeah.
So
So cool.
You do see some of the students from there. We also, there's another organization,
Big bird.
Art
Bigger bird.
for disabled adults, and they walk
Is that a
over there
grackle?
and spend-
I don't know.
You know, what's really going on here?
Is that a chimney swift tower over there?
Yes, but it doesn't have any chimney swift. It doesn't
Yeah.
have any swifts. We have a chimney. We live close to here, and we have a chimney swift tower in our yard.
Oh, right here.
Yeah, that weird looking
Yeah, it's
vertical
kinda-
box
we
thing.
Like an Eagle Scout project.
Uh-huh.
That's cool.
Yeah.
$10. Very interesting
Yeah.
stuff.
Fantastic. Thank you.
Do you know the Kyles? They're kind of famous. They live here in Austin. They have
I don't know, but I should, I guess.
They have a preserve,
Okay, cool.
and they built their house. in
Bird holder this time?
No.
Yeah, let's have Connor
Oh.
release one.
Yeah. So this one just needs to be held for a few minutes,
Okay.
and then we'll check the cotton ball that we're good.
Yeah, we're clotted.
Yep, exactly. So let's not let them
We're
go.
in cover head chat. How do you recommend getting involved with local conservation? What are your
Me?
thoughts, Joe?
Oh my gosh. Go to your local park district and see if they have anything that you can volunteer with. I started by helping with a summer nature camp. That's a great way to get started. You can, depending on how old you are, check with your local university and biology department or whatever department that might interest you. And hit them up and look for volunteer opportunities. Local Audubons are great to help and volunteer for. So
Nice.
yeah. Just sometimes all you gotta do is ask.
Yeah. And another question came in for you.
Okay.
So there's a bunch of new people that came in. So what are you using the blood for? They were wondering.
Okay, so I'm using the blood for lots of different things. And so what I'm doing right now, we're making these blood smears or blood films. So it's just a tiny drop of whole blood that I've put on here. It'll dry up super quick. We'll bring them back to the lab, stain them so that the cells pop out under the microscope. And we're going to count the different white blood cells that we find in here to give us an idea of the health of the bird.
That's cool.
So the next thing we're going to do now is I've got this instrument here, the HemoQ, that measures hemoglobin, and hemoglobin is the portion of blood that actually transports around the oxygen. So this will give us an idea if the birds are anemic. So another measure of quality. So what I'm going to do is just grab one of these little cuvettes. It already has, you see there's yellow in there, there's reagents, chemicals in there that'll help us measure. I'm just going to put my capillary tube right onto it. It'll suck up some blood. I've got some on the side, so we'll clean that up. And then I'll put it right into that little instrument, and it'll take 30 seconds or so to give a reading. So while that's going, I'm going to use this guy, the glucose meter that I just picked up at the pharmacy. And just like a diabetic would, I'm going to check the glucose level of the blood. So I just put that little strip in there, grab the capillary tube, and I just need to touch that to the side to pick up a little blood until
Nice.
it beeps, and it'll give me a measure here pretty quickly. 205, so I'll write that down on my envelope.
So, do they all run about 200 for glucose?
The birds, yeah. I had one here, the second bird, I think that was like 180, which I think is kind of low, just judging by all the other measures that I've gotten. The hemoglobin here is coming out as 18.4, which is pretty standard for an adult bird, I feel like. The chicks have much lower hemoglobin, so it's usually between 10 and 12. And then the anemic ones can be as low as 4 or 5.
Ooh.
So now I'm going to put this, the last thing I do with the blood, which arguably to me is the most important part, is I'm going to
Oh.
blood into
Nice.
this tube here. I'll use this little wipe so I'm not putting my mouth directly on
Right.
on the glass tube. And so this is a buffer. These are chemicals in here that protect the DNA and the RNA so that I can bring it back to the lab, extract DNA from here, and then sequence the DNA.
So look at how weird that blood drop
Doesn't
looks
it?
in there now.
Yeah, because it
That is cool.
coagulates right away.
It looks like
Huh.
the black stuff from Venom.
Yeah. So it's not just like a heparin solution. It's something proprietary because
Yeah.
normally you have to do these sequences pretty quick, right?
So now that they're preserved like this,
Yeah.
it's actually fine at room temp for quite some time.
Otherwise, we'd have to
Yeah.
freeze it.
Yeah.
That is so cool.
I know, it is awesome.
That makes field work so much easier than have to bring like a
Absolutely.
freezer
Especially
out
if
with you.
you're working in really remote places. This
Yeah.
is super, super helpful. Now, the capillary tubes are heparinized.
Okay. Yep.
So when we were just doing research in the Amazon on purple martins a month or two ago, we had to bring a giant canister of liquid nitrogen because we were just out in the middle of the jungle. You know,
Jesus.
we couldn't plug in.
Right.
We couldn't plug in a freezer, and if we had dry ice, that would have evaporated because we were there for about a week. But like a super duper insulated liquid nitrogen canister.
That's so cool.
It stays cold enough and it stays for long enough.
Country were you in?
In Brazil.
So should we take a look at this
Brazil.
guy and
Yeah.
see how he's
Yeah, so-
doing?
What wing was it?
It's the...
Oh my God! My
Right wing.
What is it?
trip.
I have him flipped over on his side, so it's
Yeah,
his...
yeah.
Oh, look, another sample!
First couple of trips
It's
to
his
Brazil were
left
to try and
side.
locate habitat.
So
for
it is
martins. We're
clotted
using
up
the
here.
library
Nice.
so we could capture
And
them
so
and
he
test
is
them,
ready to
and keep
go.
sampling down there.
I'll let
then we located-
Joe
Thank you.
help you out with that.
Chat. Are we gonna name this one Binky Bonky? That's Maya's new.
What am I doing?
Oh, am I releasing it? I don't know.
Yeah. Is this one clotted?
That one is ready.
We got the cotton ball out of there.
Yep.
All right. Yeah. Just go over and just
He's gonna
take
get
your.
a video of you
Okay.
too. So.
Okay.
Chat. Say bye to
Top
Binky
hand off,
Bonky.
and it'll just fly. You don't have to-
3, 2, 1. Nice.
Binky Bonky is
That's-
Maya's new thing.
She loves that name. She named her new truck Binky Bonky, and she's been trying to name things Binky Bonky. I don't get it. She's a streamer though.
I'm gonna have to bring it up in the next board.
Yeah. Oh, there's a great question that came in. What are you guys hoping to learn from these research efforts?
Yeah, so that we're hoping to learn lots of different things, but the main reason why we are here in Texas is because in 2021 we had those crazy storms that you guys are all familiar with.
Winters.
What did I say?
Winters. You said storm.
Oh yeah, the
I mean,
winter.
storms here all the time.
The late winter storms that coincided with early migration and wiped out tons of purple martins as well as other migratory birds.
Okay.
And so when that was happening, I already collaborate with Joe on lots of different things, but we got together and super quickly got lots of purple martin landlords to get together and grab those dead birds
Right.
and hang on to them. So we have a very large sample of the birds that died during the storm, and now we've come back to sample the birds that came in after those that initial round of migration. And so what we're looking at is the genetic profile, basically. So we're sequencing the genome of the birds that died versus the birds that came in afterwards, and now the second year, the birds that came back to breed, to see how the genomic profile of the population has changed as
Okay.
a result of this extreme climate event.
Ah, interesting.
So yeah, there. You know, in addition to other things, there's a genetic element of migration timing that where birds are tuned to start migration at a certain time, and with that big freeze that basically killed every single purple martin that had arrived when that storm happened.
Jesus. Yeah.
It just basically removed all of, you know, whatever those guys were tuned to show up earlier. There's still variation that will reemerge in the
Right.
population, you know, recessive genes and all that kind of thing. But there's just this really interesting genetic study to be had that, you know, you could never do a study like this. You know, you could never get approval to just
Right.
kill all the purple martins at a certain date. So, nor would we want to. But, you know, it was an opportunity to better understand purple martins. And if you know the genetics of migration timing, you can predict how much flexibility they may have for climate change,
Right.
changing
Yeah.
the time that they ought to be here. Because, you know, over eons, martins have evolved to arrive at the perfect time to have a nest and have babies. You know,
Now that's shifting
now
dramatically.
that's shifting because we got late winter storms. The timing of rains have changed, so maybe the time of year when there's lots of insects that come out because of flooding or whatever, that may have changed, and they may have evolved to where that was the perfect timing for right when the babies needed to eat a lot of bugs.
Right.
And they were perfectly tuned into that, and that might be changing. So if we know how much variation there is in that timing, we'll know how well or poorly they may be able to adapt as we continue to change the environment.
And if you can figure out their genetics and time things right, maybe this can do two things. One, you might be able to step in and help maybe change up the gorge to be more insulated, or do something to help them.
Yeah.
And then there could also be public health implications, because if you notice that there's not going to be as many insects
Yeah.
one year because the population's way down, you might need to let municipalities know, and
Yeah.
maybe there could be a spread of West Nile more than another. Like,
Exactly.
right?
Yeah, they may be so out of sync with the timing. Purple martins here in Texas and in the South, invasive red fire ants are a big problem.
Mm-hmm.
You know, they cause all kinds of agricultural problems. They're a pest to humans. I mean, you've probably gotten fire ant bites at Alveus, right? I've gotten fire ant bites. Last year there was a fire ant mound right next
fun.
to that gourd rack, and I was standing in it for a little while.
Ew.
I always wear flip-flops, so
Oh, yeah.
that was fun. But if they're out of sync with that, purple martins love it when fire ant mounds are producing flying ants. I don't know if you guys know about
I didn't know
ant
that.
biology, but at a certain time of year, ant colonies have winged ants that all take off at once, and each one of those is a new queen.
Oh, wow.
So purple martins love it because it's like dinner bells are ringing, and they just swarm and just eat as many as they can. They love to cue in on that and eat lots. And if for whatever reason the timing is off for purple martins to be here when that's happening, maybe the fire ant problem gets way worse.
And you can
in
let
Texas
you can
and
let
the South.
people know ahead of time.
Yeah.
You can get so good where you can predict
Exactly. So
that.
Um,
That's cool.
yeah, with we're trying to figure out what they eat through looking at the DNA
Oh,
and
yeah.
their diet. We already know they eat fire ants, but they presumably eat a lot more agricultural pests. You know, pests here in the garden,
Right.
pests of humans, human health. You know, pests
Like do mosquitoes?
They don't eat a ton of mosquitoes. It's a myth started by a guy who wanted to sell purple martin houses.
Oh.
So the story that they eat 10 times their weight in mosquitoes every day or something like that. That they just are not in the right place in the right
Right.
time where they eat to have mosquitoes as part of their diet. They'll eat some,
Right.
just coincidentally, but they typically will eat really high up in the sky. And mosquitoes are hanging down here low next to all the warm-blooded
Right.
mammals where they want to eat. And mosquitoes tend to come out at night. Purple martins are eating during the day, so they just are not out at the same time. If you want to get rid of mosquitoes, get bats.
Yep. We got to put it back.
Bat houses. That's what you do to get rid of mosquitoes, because they're eating at night and they're eating at the same kind of altitude
Right.
that mosquitoes are at.
So, can you see the proportion of insects that they eat in their poo, or can you just tell the species?
We could just tell the species.
Gotcha.
Yeah, the proportion you can't really get from the DNA analysis technique.
Gotcha.
That would need to be something to where we're actually looking at the fecal samples. We're looking at stomach contents.
Or I build a little tiny camera that's 1 gram as well, and you can watch what the
Perfect.
purple martins are doing.
Perfect. They're gonna wear it on their head.
Yeah.
And you see what they catch.
You can put it on their back, wide angle. Be
Okay.
perfect.
Perfect. Yeah, just like an eagle can.
Yeah.
Yeah. Well, actually, there's some studies that have been done not with purple martins, but with bluebirds and other insectivores to just take pictures every time you see them with something in their mouth.
Right.
And like with that bird we took out of the nest there earlier that had food in its mouth, you could see what they're holding, and use that as an analysis of what their diet is.
There's one last good question before I got to head out. Can you see species that rely on weather patterns going extinct out of confusion and inability to adapt to change?
Yeah, potentially. At the very least, they will need to change their ranges to survive.
Sure.
With animals that need a certain temperature, say they're in a desert, and they need things to be, a certain precipitation level or a certain temperature at critical times of year. But there's mountains nearby that, as you go up a mountain, the climate changes.
Yeah.
They may have to change their range and slowly creep up the mountain as the climate is changing. Things like that will probably
that goes far north. Yeah.
I think there's
the things. Yeah.
Is it eBird, Cornell Labs?
Yeah, I saw that.
A kind of a map prediction.
I thought that was amazing. Yeah.
Of where the ranges of different bird species are going to change.
Some expanded, some declined.
Exactly.
Yeah.
Yeah. There's more area available to them because it wasn't as cold in certain areas, and they could take advantage.
I think bald eagle ranges expanded more north.
Yeah. So there will be drastic changes, and then if you're changing the species that are in a particular area, that changes the ecosystem immensely. So, climate change is a huge risk to not only purple martins, but to humans and just the health of the planet.
What makes purple martins special?
What makes them special? The thing that really links them to humans is that where 99% of all purple martins live and reproduce in North America, they are unable to reproduce naturally in natural environments anymore. The habitat that they would have used to nest in is either removed by humans or they're outcompeted for nesting habitat by invasive species. So the only place all those purple martins in almost all of the purple martin range can the only place they can reproduce is in birdhouses that people put up in the air.
That's crazy.
So
thank y'all for coming.
Yeah, absolutely.
Yeah, thank you.
She's done. Stream's over. She's out. So there's this incredible link with humans where they've kind of adapted alongside people to tune in to where people are, to take advantage of this habitat. So humans have created the issue with purple martins, but humans can also be part of a solution to what they need. And
that's cool.
They're an important part of the ecosystem and the planet.
Yeah, you said quarter of a trillion bugs?
Quarter trillion bugs.
A year.
A year.
That's a lot.
It's a lot of bugs. A lot of bugs, and a lot of those are bugs that's it's important for us to have less of.
Yep.
And without purple martins and other insectivores, that balance is getting thrown off.
Cool. Well, guys, I've got to head back to Alveus, but all your donations, thank you so much.
Thanks, dude.
It goes to Purple Martin Conservation Association, which is Joe's the executive director of.
Sure, I'll see you all soon.
Yep. Thank you so much. That was wonderful. And hope to do some research next year at Alveus, as long as we have birds. So.
Cool, guys. Thank you for caring.
That was awesome. Well, I'm going to turn this around, wave bye to them. There's a delay. Let's all wave. Bye, chat.